happy to have him here. Sorry for that. I was just being told that the recording is in progress. So we're very happy to have Adrien. I'll just say as usual, if you guys have questions, please put them in the chat. If there's something sort of, quick and immediate, I'll sort of speak up and bring that to Adrien's attention. Otherwise, we will do the Q&A as normal afterwards. So without further ado, Adrien, please take it away.
Thank you, Scott, for the intro. Thank you for the very kind invitation. It's a very great pleasure for me to talk to you today, and it's especially actually happy and excited because this is the first time I'm presenting this story, and actually for the first time in a very long time, I like all the slides in this talk are new, so I apologize in advance if like there are small glitches here and there, but it's a totally entire new line of research for me and for the lab, and you will see why. So I spend 90% of my scientific work studying the spatial navigation system and memory formation in mice. We're using all sorts of techniques for that. But on the side, I've always been interested in also in understanding what is happening in humans, especially human epileptic patients. Of course, I don't need to tell you, and I will show you why. There are very interesting parallels between electrophysiology of the spatial navigation system, especially the hippocampus in rodents and all mammalian species, and the epileptic brain. So when I started here in 2016, as Scott was saying, I was approached by the people at the Montreal Neurological Institute saying, would you be interested in doing human electrophysiology? And I say, oh yeah, absolutely. And this is something I had done with Alain Destex, years back. It was a collaboration with the team of Sidney Cash at Harvard University, but then I say, okay, this is time for me to actually start it seriously. And today I'm gonna present to you actually the first project of resulting of this work, and I'm really looking forward to your feedback, questions. Please interrupt me whenever you want, because we're here to discuss and share these new findings, together. So. Ripple oscillations, this is in the title of my talk. This is what we're gonna talk about today mostly. And today, actually, I say in the healthy and epileptic brain, sure, but it will be mostly the epileptic brain. So what do I, doesn't work, yeah. So what do I mean by ripples? I'm sure many of you have heard about this term before, but I just want to make sure that we're on the same page. If you record the electrophysiological signals, also called local field potentials, in the hippocampus of a mammals, most likely a rat or a mouse in the lab, but it's true for actually every other mammalian species, you will see typically this pattern. Can you see my pointer? Just want to make sure. Moving? Yes, no?
Yep, we can see it.
Yeah, okay, good. So as the animal is walking, you will see a very regular oscillation called theta oscillation, totally out of the scope of this talk today. But as soon as the animal stops and is still, the pattern change like drastically, and suddenly it's like you see these very clearly clear sharp waves, negative deflection in the field. All right. So they were described actually a long time ago. The first observation dates back from the 60s. But Yuri Bujaki, during his own PhD work, really described and characterized this phenomenon. Okay, so this is a very characteristic of the hippocampal electrophysiological signals, sharp waves. And a few years later, Yuri Bujaki, him again, described the fact that on top of these sharp waves, you had like a fast oscillation riding really on the trough of this sharp wave. And this fast oscillation, he called it the ripples. They are typically in the healthy brain between 100 and 200 hertz. And they're basically, how to say, they result from the very fine temporally coordinated spiking of neuronal population in the hippocampus. All right, and they are also observed during non-rapid eye movement sleep. But again, we're not going to talk too much about it today. So how do they emerge? Like here, like it's like schematics. Not like don't take it with a grain of salt. But basically, the idea is that the sharp wave itself is a population bursts that arise from an attractor networks, meaning a network in which there is more excitatory connection more than many other places in the brain. And as soon as the animal stops, actually, the inhibitory system, the inhibitory neuromodulatory system, decrease its activity, so the systems become disinhibited, and suddenly, spontaneously, you have bursts of activity arising from like these interconnected networks. In the hippocampus, it's a CA3 region for those of you who are familiar with hippocampal anatomy that will spontaneously, show this burst of activity. So that creates a sharp wave, and the sharp wave itself recruits, downstream targets, especially in the CA1 region. And in this place, the strong activation of both excitatory cells and the inhibitory cells create this, kind of ping-pong communication between the two population, and at a very fast pace that will create this oscillation at about 150 hertz, called the ripples. All right. So that's basically very schematically what is a sharp wave ripples. And I'm not going to use sharp wave anymore in this talk, but again, it's these two phenomenon are the two faces of the same coin. It's the same phenomenon. You have a sharp wave. If this sharp wave is generated by the local network, very likely they will be associated with the ripples. So this is the what happens in the healthy brain. It starts in the hippocampus, and it has a global impact. And let me show you two examples. There are many others, so I apologize for not citing the entire literature on the field. But on the left, this is to me like a very classic paper from Nikos Lokotidis, showing so here in that paper, they managed to record, the bold response with fMRI in alert or anesthetized monkeys while recording local field potentials in the hippocampus. And what they observe is that surrounding the time of sharp waves, you had a global activation of the neocortex, right? Almost every cortical region show increase in bold response, while subcortical structures actually show decrease activity, at least decrease bold signal. All right. On the right, this is totally different types of signal, but also showing that the ripples is embedded into a global dynamics of the forebrain. This is another, to me, classic paper from Siapas and Wilson, in which they recorded local field potentials and action potentials like neuronal activity, both in the hippocampus and in the medial prefrontal cortex, which is one of the major output of the hippocampus in the brain. And what they observe is that the ripples in the hippocampus is followed by spindle, thalamocortical spindle, which is a rhythm that is spontaneously generated by thalamocortical networks. So you have a ripple in the hippocampus, and that triggered this global event in the forebrain again. So meaning that all these things are, tightly linked to each other, and the ripples does not happen in isolation in the healthy brain. All right, so that was a very short introduction about the ripples in the healthy brain. Why it's so interesting in epilepsy? Well, more than 20 years ago, it was shown that a classic model of epilepsy, which is a kainite acid injection, actually lead to the emergence of ripples, actually fast ripples, because they are faster than the healthy ripples in the limbic structure, like the hippocampus and parahippocampal structures, like the entorhinal cortex, as you can see here, and only on the side that was treated with kainite acid. And it was actually discovered more or less at the same time by the same people that the same fast oscillations were also a characteristic of the epileptic brain in humans. Okay, so using intracerebral recordings in patients, they showed that so like these, some of the same structures in these patients that that have mesiotemporal lobe epilepsy have also fast ripples. All right, so again, so you have the classic ripples here in the hippocampus, you know, around 150 hertz, and the fast ripples riding on top of what is, in this case, like a really very big shock waves, sometimes also called spike. Okay, so you have this massive population burst, and riding on top of this population burst in the epileptic brain, you have this fast ripple. All right. So here, I mean, you may not all agree, but again, I don't want to get into this debate, but for the sake of clarity in this talk, I will call fast ripples all the high frequency events, right, above 200 hertz that are associated with interictal epileptic discharge. All right, so this is very important to understand. This is not an epileptic seizure per se. This is a typical event that take place in between, sometimes actually very quite often in some patients or treated animals. And this is a very characteristic of the epileptic zone, or also called the irritative zone, meaning places in which you see these population bursts called spikes in the LFP, and on top of it, you have these fast ripples. The reason why here in this talk, I'm gonna focus on the fast ripples, it's actually a very efficient way to determine where is, you know, the time of interest in this event. Sometimes these spikes or interictal discharge can be multiphasic, quite, you know, complex, but at least if you isolate the fast ripples, you know, where likely, of course, I'm happy to discuss it, but we're very likely the neurons fire at least, you know, more than usual. And why is it interesting from a clinical perspective is that it has been suggested, even if it's still debated, that these fast ripples are markers of the epileptogenic zone, meaning that if you detect fast ripple, you have actually it provides you some insight of like where is the lesion tissue, and so that it helps, you know, for surgical. For the decision of like which brain area should be resected to treat the patient after that. Okay, so what, like, you know, these fast ripple, how are they observed? They can be observed in two different ways, and very importantly, the thinner the wire you use, actually, the higher is the resolution of the signal, and especially of the fast ripples. What do I mean by that? So what you see here is the classical depth electrode. This one is from Adtech, and this is like, you know, these depth electrodes that are implanted in usually, you know, drug-resistant patients to localize the epileptogenic zone. And you record with macro contacts that are, you know, a few millimeters wide, and that provide you with an idea of the local field potential in pretty big, you know, volume around these electrodes. And usually not at it's a very good spatial resolution when you're looking for something like the epileptogenic zone, but it's not at, you know, the resolution of this very small cortical circuits. Adtech, a company that, one of the companies that manufactured this kind of depth electrode, more than 20 years ago introduced a new version in which you have micro wires popping out of the tip of these depth electrodes. And these micro wires are much thinner, of course, much smaller than these macro contacts. All right.
I think we have a few questions, Adrien, that is not hard
Yes.
to answer at this point.
Yes.
Yes, yeah, so there were a couple questions in the chat. I was gonna leave those to the end, but I can ask a couple of them now. So there were a couple of questions about the ripples. So Karen Davis had asked, are these ripples seen in other parts of the brain, or are they associated with any particular behavior or function or a particular cell type?
Okay, so yes, actually, I forgot to say this. So the ripples, so several answers here. First of all, in the healthy brain, usually they are restricted to the hippocampal region, okay? Again, they have been observed in other places. We can discuss it. For example, in the piriform cortex, perhaps in the amygdala. But I mean, let's not discuss it. Like usually, classically, it's like, you know, it's, yeah, it's in the hippocampus. Whether there are different cell types involved, actually, it's a network level effect. And all cells somehow, or many different cell types, participate in the generation, excitatory cells and different types of inhibitory neurons. Their functional properties are very interesting, and this is something, sorry, actually I should have said it. Of course, in the healthy brain, there's been a lot of work and a lot of, you know, of studies trying to relate these sharp waves and memory formation. Okay, and this is what I've been doing myself for many years, and I'm still doing in the lab with our animal models, is to understand like what is going on during these ripples. And the theory and the idea is that during these ripples, you know, memories that are encoded in the hippocampus are replayed, and this replay facilitates the long-term formation of memories. I'm not going to say more about this because this is not something we're going to talk about today. We're going to really focus on the physiology of the ripples. All right. But thank you. I should have said it.
Awesome. And then just really quickly, there were one more quick question. So Mary Pat asked, do fast ripples also have global impact, or are they focal and sort of localized to that epileptogenic zone?
So let's keep it till the end, right? So this is the entire subject of today.
Perfect.
But this is a great question.
So yeah, I'll you keep posting questions in the chat. We'll probably get to most of those at the end, unless they're really quick questions going forward.
So here, you know, you see an examples on the right side of your screen of different signals that are recorded either on a macro contact on top here, right? So you see the spikes, which is this interictal discharge in the epileptic brain. And if you look at the signal, but if you look at it, so there's a small spike on top of it, you know, whether or not it's a fast ripple is unclear. However, now if you look at the signals you obtain from these micro wires that are right here on the tip of your electrodes, you see a very fast oscillation. Okay, this is typically a fast ripple at a much better resolution, spatial resolution that you have with the macro contacts. Okay, so here you have this inset, zoom in, and you see this very fast oscillation riding on some part of the phase of the spike, of the population spike. So now you understand why it's another, you know, illustration of why I'm insisting on this fast ripple as a marker of like a precise marker of an interictal event. It's because it's really a characteristic of the phase of the spike at which something, to me at least, interesting is happening. So we can say, well, if I detect the fast ripple, at least I have a very precise time here of what is going on, because this spike here can be of very different shapes depending on the patient, depending on the area, depending on many different things. All right. And interestingly, you know, around these years, they compare the kind of ripples, fast ripples you can detect from either macro contacts or macro or micro. It's really hard to say for a French guy. And what you see is basically these two slides, these two figures show that you observe more fast events that are fast ripples on the micro wires than on the macro wires. And the reason is that very likely, and this is what one thing we're gonna talk about today, is that the fast ripples are generated by small circuits, and the small circuits somehow may be quite independent from each other. And on the macro contact, you sum them all and the amplitude basically vanishes. So you, by summing all, you know, these small fast ripple generators, then you don't see it anymore if they are not perfectly in phase. And that's the idea. Okay, so this is a really good argument to say, well, if we want to understand what is going on very locally in the brain, and potentially down at the neuronal level, we need electrodes that have, you know, that contain micro wires. And this is what is going to give us the best resolution to understand what is going on exactly in the epileptic brain, at least at the neuronal or small cortical circuit level. All right. And so here are the big questions that we're gonna talk about today. What are the neural bases of fast ripples? Like what is going on at the level of neurons and small, cortical circuits? What is the anatomical scales of the generative circuits of fast ripples? What is the impact of these fast ripples on global networks? So they emerge perhaps very locally, but like remember what we said about the healthy ripples, they have global impact. What about these fast ripples? And what properties of fast ripples are universal, like that really you see in all patients, or in contrast, are specific to patient or specific to certain circuits? I'm not telling you that I'm gonna answer this question, but clearly this is something we should have in mind if we really want to understand and if we really want to, provide some insight or some useful ideas to clinicians. This is like the kind of dialogue we want to establish and to understand it better. So today I'm gonna present data that are recorded with totally new types of electrodes, quite similar to the one I presented before from AdTech, but it's from another company and it has like some more features in it that makes it very interesting. So this is a depth electrode. So this is something which is a few centimeters long, right? So this is the part that is going to be inserted intracranially. And you have several macro contacts on top of it. You see it in black. Okay, so these are the typical ones that are used for clinics very commonly. But in between two of these macro contacts, in this case, between the first and the second one, the very tip here, you have three bundles of micro wires that can be extruded, they pop out of this electrode by turning a knob, which is outside the skull, okay, like on the head of the patient. So you see here some more details. So it can be extruded up like by up to 2 millimeters out of this depth electrode. And actually these bundles of wires are in a tetrode configuration. What it means it's like 4 wires, extremely thin wires. They are about 20 micrometer each. Okay, they are twisted together and glued together, so that basically now we're gonna observe the local signals in the brain from 4 points that are very close from each other. And by using some triangulation, we're gonna be able to do some very interesting thing with that. So it's an extremely high spatial resolution. This is the same kind of resolution that has been used in rodent work for more than 20 years now, okay? And we know that very likely this kind of configuration will allow us to record single neurons, like activity from single neurons in the human brain. And this is here an example. So you have your macro contacts here. Typically, they will record this kind of local field potentials, which are really, coarse grain average over a large volume. Then you can also record, of course, local field potentials from, these tetrode wires. So it's like a much higher spatial resolution, and usually it provide you with like some much better signals. So this is actually, a simultaneous recording here. You see it now on the tetrode wires. And if you look in the high frequency spectrum, you can observe actually action potentials emitted from neurons in the vicinity of these tetrodes. And you see here this negative deflection, there are action potential that you hear, like you see the that you see from the neurons close by. And the fact that you're using 4 wires will allow us to discriminate between the activity of different neurons, as I'm gonna show you, all right? So these new electrodes, they are still actually, they are not approved anywhere. They were first tested in France. And especially in this in Toulouse Purpont Hospital by the team of Emmanuel Barbeau and Luc Valton. And they've been working on it for like many years now. So like a collaboration with Dixie, the French company developing these electrodes. They came up like now like with a fantastic data set of more than 50 patients implanted with the Dixie so-called hybrid electrode, hybrid because they have macro contacts as usual, but in addition of the micro wires. And Jonathan Cureau is a neurologist who did his PhD with them. recorded the first patient himself, and actually came to my lab here for his postdoc to learn, neuronal data analysis. And what I'm going to present today is the fruit of this collaboration. Also, the reason why we started to collaborate is that we have set up the same system here at the Montreal Neurological Institute, and we recorded the first patient on Tuesday. And the first patient had at least 1 neuron, and we were all very excited about it. But so too early to actually analyze. So all the data I'm going to show you today were recorded in Toulouse, in France. All right. So here's another example of the kind of signals that is observed in a human epileptic brain. So these are, the 12 signals from the 12 macro wires from 3 tetrodes from the same hybrid electrode. All right. And what you see already in this example is very interesting. So this is a raw signal, totally unfiltered. And you see the spike. Okay, so this positive deflection, perhaps actually a bit biphasic in this case, on top of which on one tetrode, so perhaps you see like the fast ripple. If you look at the filter trace in the fast ripple band, you see the fast ripple of high amplitude right here, okay? On top of this, so the spike, the fast ripple, and what you observe also, these very tiny events here, which are the action potentials of the neurons surrounding the tetrode, all right? So when do these fast ripples occur? So that remember that was my first slide. In a rodent, they occur as soon as the animal stops. And the first question we had is like, is there any relationship between the between the occurrence of these fast ripples and the behavior of the patient? So the patients were recorded for about an hour, for these tetrode recordings, and they were basically sitting or laying on the bed and asked to watch something on TV, so not super attentional demanding task. And we had video monitoring of the patient at the same time. And so the first idea we had is like, perhaps we can just track, how they move during the recording and see if there's any relationship. So to track this, when you have only one video, no markers, you can use one of these fancy new tools called Deep Lab Cut, which is, a deep net based algorithm that will track certain part of the bodies very reliably with minimal manual intervention. So here, for example, like what we've done is like an old patient, we've tracked the nose, the chin, the 2 shoulders, the 2 hands, and we trained the network on some, images, and then basically you can track the position of these body parts during the entire video. From these markers, then you can extract like a global energy, global movement value, which I plotted here. So you see here, for example, the patient was still, and suddenly at this moment, she started to move. All right. So you see a movement. You see movement also here. Sorry. So the orange arrow, you see she's here. Then she moved, and a few seconds later, now she has like crossing her arms. All right. And what you see, the ticks here are the occurrence time of fast ripples in the hippocampus. And you see that right during movements, you don't detect any. And you can look at the bigger now time resolution, much larger time resolution. Again, this is the movement in black. And as you can observe that as like when the patient here, or the subject, I should say, was moving, was active, there was no fast ripple whatsoever, all right, here and there. And even at the very fine time resolution, as you can see here, however, when standing still, and very certainly, mind wandering, a kind of unattentive, perhaps a bit sleeping, I guess, that I cannot be totally sure of, you have fast ripples. And this is here, you see across patients, 3 patients that had like video tracking, and you see this strong decrease of fast ripple rate as soon as the subject was moving. All right. So very interesting, because it really looks like what you observe in a behaving animals, but now with epileptic signals. So let's get back to this recording here. I said, so this epileptic discharge, interictal discharge, fast ripple spikes. Let's look now, interestingly, at the fast ripples recorded again on the 3 tetrodes from the same depth electrode. All right, and the thing is that they look pretty different already at this scale. If you see is that the phase relationship in this case is okay, but we have other cases it's actually totally not one-to-one. Here you see that the amplitude also is not correlated. There's some kind of fluctuation on a cycle-by-cycle basis. So it looks like, yes, they occur at the same time. However, there's some sort of, independence of these fast ripple generators. And what we know is that the distance between the tip of these tetrodes is about 2 millimeter, all right, at the tip. So very, I would say it's a bit hand wavy here, but we could, make the hypothesis that just based on this data, the generative circuits of fast ripple may be as small as 1 millimeter square of cortical tissue. It really seems that it's the case, and this is something I'm gonna try to convince you about. That these generators are extremely local. So we have now like another example. So this is one depth electrode with 3 tetrodes, right, in the posterior hippocampus. At the same time, you have another depth electrode that had only 2 tetrodes in this case in the perineal cortex. You see 2 spikes here, perhaps even the third one, and one in the perineal cortex. And if you zoom in on the second tetrode, I think writing on this spike here, you observe, as I told you, all these small deflection, the action potentials from the neurons. You use automatic algorithm in this case that will detect this waveform and cluster this spike waveform. This is called spike sorting. And the idea is that waveform of similar shape are putatively emitted by the same neurons. And at the end of your spike sorting procedure, this is what you obtain. This is from one tetrode, from this tetrode. You obtain different, in this case, 7 putative units, isolated neurons that have different waveform on the 4 different wires. Like it's again really like a triangulation of signal, and what you see on the bottom, this is the autocorrelation of spike trains, and this is usually a way to know whether your spike sorting was efficient, was good or not, because you see when you have a big dip around 0 that actually this unit had a pretty good refractory period, and this is a good sign that actually it's a good spike sorting. All right, so you see these units here, very bursty, like a lot of firing around 0. This is typically a sign that these cells are excitatory pyramidal cells, but again, not the subject today. All right, so we can record actually pretty high number of neurons from one tetrode in this case. And then you record on different tetrodes in different places in the brain, so you can show a raster plot. So each tick here is the action potential of one neuron. Neurons are organized by rows, and you see that these spikes here in the hippocampus are associated with a sudden increase in firing rate of these neurons. All right, and right after, actually, you see a period of silence, which is interesting. However, the perineal cortex doesn't seem to care that much about what is happening in the hippocampus in this case, all right? Except here, where it's a bit silent, which is interesting. So what we're gonna try to do here across patients, so there are about like 10 patients at the end in this data set in which there were, a good yield in neurons, patient in which we had also fast ripples and writing on top of interictal spikes. So there was a lot of criteria, meaning cutting really down, the data set. But with 10 patients, we were happy, and we analyzed the data set at 2 different scales in this here. We looked at local interaction, like what is the relationship between the fast ripples and the neuronal activity on the same depth electrode, on different tetrodes of the same depth electrode, but also at the global scale, meaning what is the relationship between the fast ripple somewhere in the brain and the neuronal activity somewhere else, and sometimes actually pretty remote, okay? So local and global interaction. how do we measure now the response to a fast ripple? Well, the first thing you do in this case, very standard analysis, is to compute the cross correlation between the fast ripple, the time of the fast ripple, and the spike train of a neuron, okay? And this is typically what you observe. Sorry, there should be a y-axis here called, this is firing rate, actual firing rate between 0 and 10 hertz. This is the firing rate of the neuron centered at the time of the fast ripples, okay? The problem is that different neurons in the brain have extremely different firing rate, making it very difficult sometimes to compare the response of neurons just to compare the response of neurons. So here we've normalized, the firing rates or the cross correlation in the following way. And actually, so this is something to me which is, so this is like one example of the kind of dialogue. So this is something which is extremely standard now and more standard in animal research. And this is something I've done myself, like with a lot of data from animals. And it's really always very exciting to also to start doing it with more complex data as human data. So the way you do it is like you compute the cross correlation now on a much larger time scale. Here like plus minus 5 seconds, and you smooth the spike train with a fairly large window, like Gaussian kernel of about 1 second, and that gives you the baseline. Okay, from the baseline you can also extract, the variance, which is basically based on the Poisson model. It's more or less, the standard deviation here is the square root of the baseline. On top of that, you also smooth the cross correlation with a much narrower, finer kernel, just to smooth out all the noise. All right. So this is what you obtain in orange. And if you take this cross correlation, smooth cross correlation, you zoom in, and now you can actually do a local z-score of your data, meaning that for each time being, you determine, how many z above baseline you are. And in this case, now you say that this cross correlation at this peak here is no more 10 hertz, is like 3z above the expected baseline if there was no interaction between the fast ripple and the spike train. All right. And you can do it for every single neuron, which have like very different firing rates. So this one here at most is 5 hertz. This one is like up to 25 hertz. But you see that now when you normalize, the z values becomes much easier to compare with each other. And you see that they have very different profile, very different responses in relation to the timing of fast ripples. All right. So now what is happening locally? So what you see here, so this is what I showed in the previous slide, one cross correlation, normalized cross correlation of one cell as a function of it relative to the timing of the fast ripple on the same depth electrode, right? So it's a local interaction in this case. And this is for all the neurons that were recorded in many different patients, at least 6 or 7 in this case, from like, as a function of time, like of the fast ripples detected on the same depth electrode. And what you observe is like a majority of neurons fire very strongly right at the time of the fast ripple. Okay, so the fast ripple is associated with the transient barrage of excitation of neuronal activity in the local network. Interestingly, also you see that it's followed by a period of inhibition. Okay, now what's happening at the global scale? So now you see it's actually a bit noisier. Of course, the amplitude of the effect is lower, but however, like this color scale here, just note is the same, right? So for the sake of clarity, it's the same color scale. And you see different examples on the right-hand side in which you have neurons that do not fire at all during the fast ripples that was detected on another depth electrode, which sometimes is centimeter away, okay, in the brain. However, it was followed by inhibition, silencing of this neuron. All right, so you have two pretty good examples in this case. And if you do the average, you see that, it's more or less there's nothing here around before 0. And right after 0, you have a dip on, again, it is an average of all these neurons that are recorded on another depth electrode than the ones in which you see fast ripples. This is some statistics now, if you want to be convinced. So if you look at, the average activation of the neurons, like on the neuron by neuron basis, around the time of fast ripples, if you look at local interaction, it's of course extremely highly significant, and you have strong depolarization of the neurons. However, at the global scale, there's no modulation. All right. Now, if you look at this inhibition, which is post-fast ripple, after the fast ripple, you see that, of course, the inhibition local network is stronger. However, it's still highly significant for the neurons that are, even for neurons that are centimeters away. All right. Okay. So first, let's get back to this idea of like the generators of the fast ripples are local. What happened now exactly on the different tetrodes of the same depth electrodes? This is an example from one patient in which you see two depth electrodes and two depth electrodes with two tetrodes on which we had more like 4 neurons or more. And so 0 here, this is the fast ripple from depth electrode CP, and here this is fast ripples detected on the TBP depth electrode. All right, and 1 and 2 are two tetrodes from the same depth electrode. And you see here, actually, it's pretty interesting that the neuronal response to the fast ripple and the 0 here and there, it's the same event. It's actually pretty different. Even if these are two tetrodes like that are only 2 millimeters apart at most. All right. And again, here you see a pretty different response between the neurons on this tetrode and the neurons on that tetrode. And this is something we've seen over and over. This is another patient, two tetrodes, you see, so 0 again, this is the same time, you see activation there, you see almost no activation, but actually strong inhibition after. This is another tetrode, now we have stronger activation, and we've seen it over and over. What does it mean? Like, how do you quantify this? It's pretty complicated. Of course, the data set is limited. We wish we had like a much larger data set, but we do whatever we can with what we have. And here you see basically the fast ripple peak rate of the neurons, again in Z-score, for the different tetrodes of different depth electrode from different patients. All right, and what you see is actually extremely variable from tetrode to tetrode. So this is something that is definitely not universal, but which is patient and certainly circuit specific. So you can do an ANOVA or actually a non-parametric version of ANOVA, like cross-cut Wallis, and you say like, does it basically, do these peak rates come from the same distribution? Absolutely not, meaning that there's a huge variability. There's more variability across tetrode than within tetrodes. Okay, so so let me summarize a bit like what I've been saying so far. So tetrodes in the same epileptogenic zone do not show the same ripples. Again, so you see this ripple here is not exactly the same, and not even the same excitation of neuronal activity with the fast ripple, as you can see, for example, for these two tetrodes from the same depth electrode. So these provide even more arguments saying that the generative circuits are certainly about 1 millimeter squared. Okay, and this is interesting because this is reminding me actually of my first work with human data. There are many other work. I'm shamelessly, just talk about my own work here, but there is a lot of anatomical work saying exactly the same thing, even other electrophysiological work. But like back in the days when I was a young postdoc, we analyzed this data recording from a Utah array. So at Harvard, Massachusetts General Hospital by St. Cash, and so we had neurons on this 10 by 10 grid of electrodes, okay, that you can see here. And if you take one seed, meaning one neuron recorded on this electrode, we ask the question is like, what is the correlation of this activity of this neuron with all other neurons that were recorded on a different location? And so these lines indicate, the pairwise correlation, and the color of the line indicates the strength of this correlation, of the pairwise correlation of these neurons. And perhaps you can see that it seems that there is a spatial relationship here, that the long lines tend to be darker than the short lines, meaning that the correlation is restricted anatomically speaking. And this is exactly what we observe at the population level. So this is correlation, sorry, this is missing as a function of distance. And you see this beautiful exponential decay. And with some statistics, you say that, well, the spatial scale of this exponential decay is typically about 1 millimeter. So we observe exactly the same thing. This was not actually in the epileptogenic zone. This is a characteristic of the cortex in which, in the neuronal, in this case, actually, they were pyramidal cells. They connect with each other, they coordinate each other in an anatomical scale of about 1 millimeter, all right? And interestingly, this is the same thing we observe with the generators of fast ripples. So you remember I showed you that following fast ripples, there is an inhibition. And this is very interesting also to me. And we wanted to know more about this. So one thing we observe is that the duration of this inhibition differ from one neuron to the next. And it was clearly not the same across the data set. So the first thing we try to say, how can we know, how can we measure, quantify how long this inhibition lasts? So this is, 0 here, the time from the fast ripple, you have increase, this is in a local network, then you have decrease and goes back to baseline. So what we did actually is something, I would say, quite simple. So we just fitted a sigmoid from the dip here, from the trough to the baseline. And you say, well, the time of maximum slope in your sigmoid here, this is what we call the neuronal recovery time. Okay, so this is how long it takes for the neurons to go back to baseline. Okay, and you see for these two neurons is actually quite different. And guess what? Actually, it's very different also across different tetrodes. So these are, again, 3 different patients. You see 2 tetrodes from the same depth electrode. And look, I hope you can appreciate the fact that even on 2 tetrodes from the same depth electrode, actually this post-fast ripple inhibition is quite different. And there is much less variance within tetrodes, for example, here, there, than across tetrodes. Okay, and this is very well captured by statistics. This is, the distribution of neuronal recovery time. Okay, so this time here, so pretty broad between 0.2 and 0.8. Sorry, but not millisecond here, 0.8 seconds, up to 800 milliseconds. And you see actually all this variance that is clearly way bigger, way higher across tetrodes, again, than within tetrodes. Okay, so that was very interesting to us. So clearly there's something here that is, again, not universal, but patient and circuit specific that we do not understand. However, even if we don't understand this, what does it mean? And what can be actually, the role of this inhibition? So again, what we quantified here, we call it the neuronal recovery time. And for a given tetrode, we would call the neuronal recovery time the average of, this recovery time for all the neurons recording on the same tetrode. We will call also the fast ripple recovery time. That's something we will extract from the inter-fast ripple interval. So you take all the fast ripples you observed, you look at the inter-fast ripple interval, you take the median, and you say, well, actually, this is the same depth electrode here, CP in patient AAV20, and you see that the fast ripple in this case tend to have to be reinstated after 1 second. And you see that the neurons tend to be reinstated after about 800 milliseconds. All right. So if you look across patients now that had, sufficient number of neurons that has fast ripples, you have a lot of conditions. At the end, you end up with only 3 patients, but this is as good as it can get. What you see is that the neuronal recovery time and the fast ripple recovery time are of the same order of magnitude. Okay, so there's no like 2 orders of magnitude difference. So these times are comparable with each other. However, interestingly, the neuronal recovery time was always slightly shorter than the fast ripple recovery time. And what it means, we think, and okay, so this is quite speculative at this point, is that this post-fast ripple inhibition in local network is a way to actually prevent the occurrence of a new fast ripple, at least in a few hundreds milliseconds. Okay. Cool. So this is very likely the inhibitory network, taking over and making sure that the activity is dampened and that this fast ripple is not, does not explode into more critical activity. But it also may play a role in the occurrence of the propagation of fast ripple in global networks. So remember, this is what I showed you before. So we try to actually relate, this inhibition, the post-fast ripple inhibition, and the location of the electrodes, the relative location of the electrode, like where was the fast ripple detected, where was the neuron recorded. And it was hard, because of course the data set is also limited. We don't have all possible, combination. So we try to split it at least in few conditions. First thing we say, is there a difference when, the fast ripple and the neurons were ipsilateral or contralateral? No, does not depend. Sometimes you see actually post-fast ripple inhibition even contralaterally, okay? And there is no difference between these two distributions here. Does it depend on where the neuron is? Like, what is the status of the cortical circuit in which the neuron was recorded? No, it's actually, you see inhibition if the neurons was in the epileptic zone, in the irritative zone, meaning it's not the seizure onset zone, but you do see spikes, interictal spikes. And you will also see in healthy tissue, in tissue in which there were tetrodes because, the neurologist thought it could be a candidate area for epileptic locus, which it was not at the end. There was no spike, there was nothing. And still we see this neuronal inhibition after the fast ripples. All right. And as a control to make sure that you believe me, is that if you do the same at now for at time 0, at time the fast ripple, there's nothing whatsoever. So there's no difference across, nothing is significant. There's a slight difference perhaps here because in the irritative zone, it was slightly negative actually at time of fast ripple. Okay, so nothing is happening at time of fast ripple, very unlike what we observe with healthy ripples actually in rodents, for example. However, following the fast ripple, we see this inhibition in very global networks. So let me summarize all this. We have fast ripples that are associated with local barrage of neuronal excitation in small cortical circuits. Fast ripples are followed by strong inhibition in the degenerative circuits. What is the origin of this? We don't know. Fast ripples are followed by inhibition in remote brain areas, and perhaps that prevent propagation of this epileptic activity. So let me actually present you 2 last slides, which to open the discussion. The first one is about the origin of this inhibition. This is something, again, some part of my own work, but there are many other studies on the subject. But something we managed to do in the Bujak lab back in the days is to actually do intracellular recording in a behaving animal, in behaving mouse. Okay, so you record local field potential, you have the ripples, it's a healthy ripples, and this is VM, like voltage membrane potential of a single cell as the mouse is moving around or sleeping. And what we observe is that following the ripples, there was like massive hyperpolarization of the membrane potential. All right. And interestingly, if you inject current, so basically you're testing the membrane potential at different voltage here, you observe that this hyperpolarization following the ripples here reverses potential at around 100 millivolts, which is a potassium channel potential reversal. So this inhibition very likely is depending on potassium, not chloride. And it means that it's very likely GABA B if you're interested. However, it doesn't answer the question is like, why is it of different duration and different strengths, even in very local networks on the same depth electrodes? Like, is it because the tissue are different? Is it because they're affected in different ways? To me, these are the open questions that are, very interesting for the future research. Also, like, what about this global interaction? I told you, ripples in a healthy brain are followed by spindles. What we observe is that actually during spindles, which are thalamocortical rhythm, inhibition is strongly recruited in the cortex. And the cortex here, the sharpwave ripples here, that was the old way of calling them for me, become, actually, sorry, the legend has disappeared. So this is during spindles on gray and outside ripples. And what you see is that during spindles, the cortex, in this case, it's the cortex, becomes unresponsive to the ripples. So perhaps the inhibition we observe, because we don't see anything at the time of stress ripple, but only after, it's not a direct relationship, but it perhaps goes through the subcortical structures, such as the thalamus, and then inhibit global networks this way. So let me, this is the lab. What I presented today is the work mostly of Jonathan Cureau, who did his postdoc. There are many other people in the lab. So of course, this was like an old picture, pre-pandemic area. Thank you again for having me today. And I'd be happy to take any question. Thank you.
Awesome. Thank you so much, Adrien. So there are a few questions in the chat that were sort of lingering before we open things up. Karen, I know you had asked a couple of questions in the chat. Did you want to maybe verbally ask sort of a conglomeration of those questions?
Hi, thanks very much. Great talk. Actually, I think I was just jumping the gun a bit with some of my earlier questions that you ended up addressing most of those things after. I think one thing I missed if you said it or not, that was in the back of my mind from back in, it's been about 15 years since I did electrophysiology, but I was thinking a lot about the bursting cells that we used to record from, seen in a lot of patients, different parts of the brain, and that they were, that they were attributed in some cases to calcium spiking mechanisms. So I'd wondered if this has any relevance to these kinds of ripples.
Okay, so thank you. That's actually a pretty good question, a really good question, because the study we did intracellularly in mice, this was one of the questions we had. And what I can tell, so the question is whether the ripple generation depends on calcium currents. If you record like thalamic cells, I'm just summarizing so for people who are not familiar with this kind of literature, there are low threshold spikes processes, which are super interesting. It's like if you hyperpolarize a neuron, that actually deactivates some currents, calcium currents, famous one is called IT, that will actually then depolarize the neurons and create a burst of activity. So you can create activity from inhibition. However, it seems that the ripples itself is not dependent on calcium currents. So during ripples, it's not real, like what is observed are not bursts. At least here I'm talking about the healthy ripples in the hippocampus of a mouse. So whether it's the case in cortical epileptic tissue, then it's actually a very interesting question. So indeed, this is something we should look at. Then the question is like, how do we address it with extracellular recordings? Okay, so we have no access to the intracellular mechanisms here, of course, but there are two things perhaps we should look into. One is whether action potentials are emitted at a very short interspike interval, meaning less than 10 milliseconds, which is characteristic of a burst. All right, perhaps we should look into it. Like, do we see a lot of these spikes occurring, like 2, 3, 4 spikes perhaps at a very short interspike interval? The second thing, which is characteristic of a burst, is that the action potentials changes shape during the burst, and that's something very interesting. So the spikes become broader and smaller in amplitude during a burst for different reasons, and this is something we could look into also.
Also the interspike interval changes with successive spikes within the burst, which so extracellular recordings, I guess that's a signature of a calcium spike extracellularly.
Yeah,
Yeah,
absolutely.
thanks. That's really interesting.
Awesome. So just looking at the chat, Mary Pat had a question about epileptogenic and non-epileptogenic hippocampus. I'm assuming that because this is all human work, obviously you only have access to patients with epilepsy, so there's sort of no ability to do a sort of control study for non-epileptogenic.
So I'm going to jump in, Scott, because all of these
Yes,
studies
please.
have controls, right? Because eventually we put a lot of wires in people that end up not showing anything. So we do actually have control tissue, quote unquote, or at least what we think is. And one might argue it's the best situation of all because all of the tissue has the same drugs on board, right? So you've taken away a lot of other confounders in there. But I'm really, I loved your talk. Thanks, really interesting. I'm a neuropsychologist who deals with epilepsy patients all the time and tries to understand what's going on in our surgical rounds. So this is really very helpful for me. And I was trying to put together some of what you were talking about in terms of the inhibition and seizure propagation. So maybe one of the first questions has to do with, again, I know you have a very limited sample, but from your own work or others, how does the presence or occurrence of ripples relate to subsequent seizures?
Okay, so there's a lot of work that has been done. I think the early tendency in this field, so here I just want to make a disclaimer, first of all, that take anything that I'm saying with a big grain of salt, because I am not, I don't consider myself yet, at least, an expert in epilepsy, so this is like something I've been learning a lot, but I'm still not totally on top of the game. But what I can say is that for a long time, I would say, 2000s, people were, there was accumulating evidence that yes, there was a very strong relationship between tissues showing fast ripples and the fact that there were a seizure onset zone or epileptic zone. Then there's been some studies, even a couple of years ago, saying, well, perhaps not that much. If you look at just high frequency oscillation, but not necessarily this fast ripple, and the fact that there's a spike, actually, it's a better predictor of the outcome of the surgery, meaning angle 1, 2, whether the patient is gonna recover. And then there's been like a cross, institution a study saying, well, come on, yes, there is a relationship, meaning that if you identify the epileptogenic zone just with the fact that they generate fast ripple, it's actually a pretty good predictor of the final outcome. So I would say very, carefully that it is still a debated issue. It's not black or white, but clearly there's a relationship.
Sure.
I don't feel more comfortable. Like I don't feel comfortable.
No, thanks for that, because I don't know the answer to that, and I agree that the literature goes back and forth. But if I can just a quick follow up in terms of again understanding this idea about how it may the inhibition may be related to propagation, because at least from a simple-minded idea, one would think that it should scale with where you generally find propagation pathways to be, right? So some completely unrelated place in contralateral hemisphere, it seems unlikely. And I thought your idea about maybe, being conveyed through thalamocortical interactions was interesting. But obviously we don't have a lot of electrodes in the thalamus in most of these patients to be able to sort that out. So I was just wondering if you had any kind of further ideas about that.
I wish I could answer this question so much. Actually, I spent time myself saying, here's like the data set we have. You can imagine we have like few tens of pairs of recorded location. And I was like, does it match, for example, trying to scale it to MNI coordinates and say, does it match what we know from the connectivity in the brain? But the data set is too limited. It's too limited. So when I show, that there is no difference EPC or contralateral, don't get me wrong. So we really split, we tried to find, you know, the easiest way to split the data set only in 2 or 3 at most, because we cannot do much more than this.
Right.
Or then it's becoming like totally undersampled. But when you split in 2, even the contralateral, the location of the electrodes, like very often are not totally independent. It's like, you know, you have posterior hippocampus left, posterior hippocampus right. And in this case, only one of the 2 may be in the epileptogenic zone. However, you'll see inhibition contralaterally, but we know there's like big bundle of fibers between the 2 areas. So there are, even if I say contralateral, I was not implying that it's a totally independent structure. Actually, it still can be very related to the site of the fast ripple generator.
Right. Okay,
Yeah.
thanks very much.
But just like to conclude this, also, I did not show the full complexity. There are neurons, there are like pairs of recordings in which we don't see the inhibition. So I know it's not everywhere.
Right.
I know this. I know it from the data set. I know it's not everywhere. It's very strong in certain cases. Sometimes it's inexistent. I'm just saying it's not universal. So there must be a relationship with connectivity. However, we will need 50, 70 patients, I hope, to answer this question. And I'd love to answer this question.
Okay, thank you.
Thanks for jumping in, Mary Pat. I should know from experience that it's impossible to accurately translate the question from text. So I apologize for not.
Well, especially once we've done more elaborate thinking after the question was posed. So, but thanks.
Exactly. So before we get to the 2 hands that were raised, I know Bill Hutchinson had a sort of interesting question in the chat. Did you want to ask that?
Sure. It was just a general methodological question. If you've tried or thought of using microstimulation to sort of lead on from Karen's question, the bursting cells in thalamus that you referred to, if you pass a short current down, you get a very stereotypical response of 3 quick bursts and then a long inhibition before they return to their spontaneous firing rate. So we do a lot of characterization of the different kinds of neurons by microstimulation, either through the same or an adjacent microwire, for example. So just wondering if you had those capabilities, or if you're looking to observe that in the future to see if this sort of association between the ripple and the inhibition is also true when you sort of induce it.
A great question. Absolutely. So these patients were also stimulated. However, they were not stimulated at the same time they were recorded. I mean, the tetrode microwires were recorded, right? So we have to understand here is that usually you have a clinical system for recording that only acquired the data from the macro contacts at a low sampling rate, about 2000 hertz. And at some point you plug the micro tetrode wires on another system that runs in parallel that acquires the data up to 30 kilohertz. But that doesn't run continuous. Usually, you do it for 1-2 hours, perhaps more. But when you do the stimulation, you don't do the 2 at the same time. Perhaps it will be done in the future. Here, the MNI, yes, we do stimulation also in patients. This is done by the neurologist here, Dr. Adrien Peyrache. She does a lot of stimulation. This is ongoing project that she has. That's something we're trying to collaborate on also. Of course, it will, like what you suggested is super interesting. Like if we had neuronal recordings, try to relate the response of the neurons at different location with the stimulation, that, yes, of course. It's like, I wish.
Okay, very nice talk. Thank you very much.
Oh, thank you.
Great. So we can go through some of the hands that are raised now. I think Francis, you were up first.
Okay, thank you. Really nice talk. Really enjoyed it. And my question is sort of a straightforward, quick one, I think. Is you're, of course, I really like sort of the global local inhibition. And so my question is, and the reason I like it, of course, is because as you're probably more than aware of Silva and Williams' work with, the piece of tissue, right? That you can get the theta oscillation spontaneously. So you have this sort of global local inhibition. And so I guess what I was wondering if you could speculate on different inhibitory cell types contributing to the local versus global. Like, do you think sort of the global one could be these sort of long range, these inhibitory cells that are known to have long range contributions? Or, I mean, it's a speculation, but I mean, from work you've done in do you wish to speculate?
It's possible. Of
Yeah.
course, they are like, a subclass of long-range inhibitory neurons in the cortex. They have not been characterized at all in primates, as far as I can tell, only in rodents. Do they really exist in primates? Very likely. That will be amazing, yes, if this was the case. I don't think actually there's only one pathway. As usual, it's a combination of different pathways. This is why, I suggested at the end that it could go through the thalamus. It's one possibility. If it goes through the thalamus, though, then it's a bit easier because we know a lot about thalamocortical, feed-forward inhibition to the thalamus. Thalamocortical neurons contact a lot fast-packing, basket cells in the cortex. So through the thalamus, you can induce, strong inhibition. Yes, it's possible. The long
I mean,
range.
like, is there an experiment you could imagine designing to sort of separate these two possibilities? Of course not the inhibitor cell type, but the fact that could it be sort of, long range site hippocampal interneurons, rather than through the thalamus, like, yeah.
So
Yeah.
so the very long-term goal of this project is to indeed understand, the contribution of different cell types to neuron, like the generation of fast ripples and the propagation or not of these epileptic patterns. But that to this end, we need to record, many patients and relate, firing characteristic in terms of spike train and waveforms to what we know from genetically labeled data in animals models. So this is what we're doing right now. This is the goal of all this. But even in mice, even in rats, these long-range inhibitory neurons are not very well characterized. They're the only one we know is, the classic one is from CA1 to the medial septum, for example. There are others, of course.
Yeah. Okay.
Yeah, I think I'm pretty sure that there's been an Ammonier who showed, long-range inhibitory neurons between the two entorhinal cortices.
Yeah. No, that's fine. I mean,
Possibly, yes. I mean, yes. We can speculate. I let you speculate. I say, of course, it's possible.
Yeah. No, I mean, but just so you have
like,
that, yeah.
But thinking of an experiment, meaning it's like it's not an experiment. It's like analyzing data, right?
Yeah.
Or like the microstellation related to the previous question. Perhaps microstellation could tell us, but can we really disentangle, the contribution of indirect pathways through direct pathway? I don't know. Perhaps.
Okay. All right. Okay. Thank you.
Thanks.
Great. Julio. Julio, are you there? There you are.
Yeah. Hi. So I also wrote the question in the chat, and as I'm not I never worked with ripples, so I'm not an expert of that. So my question is, bottom line, why do we have fast ripples? So which is the mechanism that make them fast with respect to these standard ripples?
Yeah, that's a great
Okay,
question.
you have a question from me later as well, all right?
Okay. That's a great question. I can't tell that I know exactly why, but here's my gut feeling is, if like the schematics I presented in my very first slide is that first, this whole starts from population burst from, excitatory connection between a local group of neurons. And typically in a healthy brain, this is what you will see in the CH3 region of the hippocampus. In CH3, you have much more excitatory connection than anywhere else in the cortex. Excitatory connection in the cortex, local network, you have to know this is something we cannot repeat enough, is extremely rare. Like we all have like in mind, a model of the brain in which neurons communicate with each other, but they don't do it much through excitatory connection. Of course they are, but it's extremely rare. And one reason is that if there were too many of them, then it will actually become epileptic. So in the epileptic tissue, for different reasons, these connections are potentiated, or the inhibition actually has less control over the excitatory, communication between neurons. So what does it make faster? Actually, you have a broad range of frequencies. Sometimes it's actually looking like a regular healthy ripples. Sometimes you have extremely fast events. So the question is, you have the burst of activity. The burst emerge from, the excitatory barrage, the excitatory, reverberation within the local networks. But this excitatory reverberation is not coordinated in time. It doesn't generate an oscillation. What generates the oscillation is the relationship with the inhibitory network. If the inhibitory network itself is affected in epileptic tissue, it will be less, how to say, it will be worse at coordinating the activity and creating like a nice looking ripple. So when we say, sometimes very fast ripples, if you look at the signal, it looks less and less a nice oscillation. It becomes like really, very noisy. It's like more burst of action potentials, okay? So actually some people argue that above 200 Hertz, it's actually less and less an oscillation. The reason is why is that if you assume that the ripple itself arises from the communication, the synaptic communication between excitatory and inhibitory neurons, you cannot create something that is much faster than 200 Hertz. This is 200 Hertz, this is 5 milliseconds. This is the time it takes for an action potential going from the excitatory cells to the inhibitory cells, and the inhibitory cells in return to inhibit the excitatory cells and to create the oscillation. So fast means that it's quite disorganized at some point. But the one we've seen actually so far myself in this patient in the data set, they actually look pretty like good, like regular ripples, like around 200 Hertz, not much faster. So I would say faster ripples, even more affected tissue, or at least affected inhibition. If you suppress inhibition in the hippocampus of an animal, actually the ripple starts looking like a fast ripple in an epileptic animal model or a patient. All right.
Yeah. Thanks so much.
Long answer.
Moritz.
No. I think before me. Hi. Ciao, Adrien.
Hey. How you doing?
Yeah, I know. It's surprising. Yeah, I'm reaching you out there. I am coming to Canada as well. So,
Yeah.
But we'll talk later. I think before me there is Itai. So I will leave the stage to Itai, and then Scott, you can let me ask the question.
Okay.
Well, yeah, Itai, I think he put his hand down. I'm not sure if Itai, your question got answered, or do you want to? I think Itai might have had to step out.
Okay. So
yeah, Maurizio, go ahead.
I'm back. So Adrien, I have a couple of questions. So the first question is maybe a provocative one. So I was very intrigued by the fact that you said that you look at the shape, perhaps, of the autocorrelation function, and you can, en passant, say that you're looking at excitatory or, I assume, inhibitory cells. So is there such a thing as ripples only from inhibitory neurons?
No. So yeah, I say it last song because of course, like I would say, one natural question here from this data set is that do we see any difference between putative excitatory and putative inhibitory neurons? I wish I can answer, but again, to me, the feeling is that the data set is too limited. If we start splitting it again, we won't see much difference. The reason why also I know, and the problem is that there was some problem with the filtering of the waveform, making it not as easy as usual to separate neurons based on the waveforms. And I require a lot of work actually to get back to the raw data and re-extract the waveform in the proper way. However, we know also that, sorry? No. That's the echo. But what we know from the campus is that the majority of inhibitory neurons will just behave like excitatory neurons. At the time resolution of the sharp wave, meaning they will be activated. If you then look finally at the resolution of the ripple itself on a cycle by cycle basis, then you will see a difference because excitation and inhibition correspond to the two phases of the ripple. But we will never have sufficient data but in this data set, we don't have the resolution to observe, to look at this. We can try, but it won't provide us with much like much useful information, I guess. So again, later, we're building this data set. Patients are still being recorded in Toulouse, now here in Montreal. And when we'll have like sufficiently big data set, this is exactly the kind of questions we will look into. But yeah, basically mostly all neurons are activated by this. Then they finally at a fine time scale, not. It's different.
And another thing is how stable in time are the ripples with respect to the location you are looking at them?
So the ripple is actually pretty stable. I mean, once your electrodes is in place, you have a pretty good stability of the electrodes, and many of these patients are recorded in 1 and 2 weeks with these electrodes because this is the time it takes for them to have seizures. And basically, they stop medication, anti-seizure medication right after the surgery, and they wait for the patient to have seizures so that they can identify the location of the epileptogenic zone. Then the question is the stability of the neuronal recordings. That is actually worse, of course. My own experience is that right after the surgery, like the day after, you don't see much, so the tissue must be affected. You start seeing spikes in the days following this, but then it decays. Very likely because you have micro movement from the wires in the tissue that create some damage. Again, I want to stress out here is that these hybrid electrodes are very actually the level of invasiveness is extremely limited compared to what is done with the depth electrodes themselves. Meaning we're talking about like 3 little micro wires popping out of the electrode. So it doesn't do much more damage than what is done already just for these surgeries. But still, locally, if you have vibration, that will affect the tissue and that will affect the neuronal.
And so connecting, and then I close, sorry if I connect to what Mary Pat was suggesting before, no? Because I was very intrigued by this idea that you observe this global inhibition, right, on the fast ripple. So and at the beginning of the talk, you said we can look at fast ripples as local epileptogenic markers of the site. So what if instead the ripple is just a proxy for something else, which is a ping-ponging in between a double inhibition in some other areas that creates fast transient moments of disinhibition and therefore ensue in the ripple that you observe. Understand what?
Yes, I understand. However, it's an interesting possibility. Even in the healthy brain, it's very likely that the ripples is preceded by activity elsewhere. So it's not entirely spontaneous. There's some stochastic fluctuation elsewhere, especially in the thalamus. So this is something we are working on in the lab, and we published something actually a year ago showing that there were some thalamic nuclei, for example, whose activation precedes the occurrence of ripples in the hippocampus. Is it the case here? Actually, this is something we're gonna test in animal models of fast ripples, recording in different locations. Yes.
Nice.
Is there another seed in the cortex, perhaps something in an area which is even more epileptogenic? That I doubt. Because what you see is really, the fast ripple, there's really the fast ripple and the increase in firing is really concomitant with each other. And it really seems that it's locally generated, very locally generated. All right. But yeah, these are many open questions that needs to be addressed anyway. Yeah.
Well, thanks. Awesome talk. Very nice to see you.
Yeah.
So I do want to ask one quick question before we wrap up, which is obviously all this data is from the patients prior to the surgical intervention, the resective surgery. Yeah. Do you have any access to data from these patients post-surgery? Like, are you able to do any experiments on the resected tissue or monitor them afterwards? Because I asked that question because some of the stuff that we do in Taufik's lab is sort of the inverse, where we do a lot of the sort of single cell electrophysiology on the tissue extracted after the fact. But we don't have this level of detailed, analysis of the brain with the IEGs, prior to the fact. Have you done anything to sort of, do you have the capability to try to start bridging that divide?
So first, like nothing like this was done with the data set I presented. So this is not something you're doing in Toulouse anyway. Of course, you can relate it perhaps to some more high level observations, like, the outcome of the surgery, whether the patient becomes seizure free, many things that of course are tracked and are, available in the data set. However, nothing like, slice work on the resected tissue yet. Do we have the capability of doing it here at Montreal Neurological Institute? Yes. And this is definitely something that are in the, that we're planning in the long term. This is something they're doing, for example, right now, not in epileptic tissue resection, but in tumor resection. They do a lot of work, they do a lot of, single cell RNA sequencing, for example, trying to describe, the neuronal types in the tumor and the tissue surrounding the tumor. So we can totally think of doing the kind of same work with epileptic tissue. Yeah, not doing it yet, but of course would be a great great thing to try.
Yeah, I'm just thinking, especially with the inhibition elements that you're talking about, it would be very cool to be able to, to characterize, some cortical slice relevant, even if it's not the exact cortical slice that you're measuring from, to perhaps see how, the inhibitory populations are affected by epilepsy, these sort of long-term projections that you were talking about. That could be something that, either single cell electrophysiology or even something like transcriptomic, analysis, from, even, sort of later tissue, could be something cool to probe that.
I think, so it's also along the lines of previous question that were asked, whether like what kind of other, experiments we can do to probe, the kind of connectivity. Of course, one thing I strongly believe in is to combine these recordings with all the techniques, especially non-invasive techniques. Of course, we all think of, fMRI. That's complicated because actually these electrodes are not MRI compatible. We can do it with MEG, perhaps. This is something that is done right now at the MNI, and also and MEG recordings at the same time as depth recording. And there are all the techniques that are coming up, super fancy, exciting techniques such as functional ultrasound that we will soon start doing here. We just had actually a CFI grant to buy one equipment, so we'll see if this is the kind of thing we can do. Yeah, exciting times.
Yeah, awesome. So I think it's 11:30, so I think we'll cut it off there. Adrien, thank you so much for a wonderful talk and for answering all the questions. So thank you again. We will see all of you at the next KCN event in the coming months. Bye, everyone.
Bye.